Thank you for the introduction, Dr. McMahon. We are, of course, from the McMahon Lab, and we're presenting our research on using quantum neural networks, quantum vision transformers, and the mathematical morphological reconstruction algorithms for brain tumor detection. And I'm Tiffany Liu.
I'm Riddhi Sharma.
And I'm Shravansh Bansal.
Okay. Brain tumors are the uncontrollable growth of cells that form an abnormal mass of tissue, appearing in the glioma, meningioma, and pituitary areas of the brain. And they can be either benign, meaning they're non-cancerous, or malignant, meaning they are cancerous. They also come along with a variety of symptoms, including fluid blocks, brain pressure, internal bleeding, and loss of balance, vision, and memory. The rising rate of undiagnosed and inaccurate brain tumors is affected by current approaches, which cannot detect the formation of these tissues early enough. In fact, the National Brain Tumor Society states that 1 million Americans live with a brain tumor, where 94,390 people get a diagnosis, which includes a 35.7% relative survival rate in 2019 alone. Our team is approaching this inefficiency by investigating mathematical morphological reconstruction algorithms, quantum convolutional neural networks, and the quantum vision transformers to result in an early stage detection model with Skimmage, TensorFlow, OpenCV2, and Qiskit. So currently, the most common form of tumor detection is done manually by radiologists, or MRI, with MRI scans or magnetic resonance imaging. Oh, sorry, the slide changed. Okay, and this poses a few issues. So as the amount of data has been increasing, it's become a lot more inefficient and tedious to manually sort through all of this data. Further, this form of detection relies heavily on the experience of radiologists, which may be prone to error. Also, many areas don't have the resources or the doctors necessary for this. So, in an attempt to address these issues while accounting for accuracy and speed, there have been a lot of recent and successful developments in deep learning approaches, specifically with the CNN algorithm. And that led to the basis of our research, which is to see if the MMR and the QNN and QVT algorithms can perform better and see if introducing this quantum aspect would give us any advantage. So first, we started by collecting a dataset of 1,711 tumors and 1,856 non-tumors MRI images, which is split into an 80 to 20 training to testing ratio, filled with various tumor shapes, types, and orientations. To start off the process, the images go through pre-processing, which is done in a skull stripping algorithm to isolate the brain. The output of the skull stripping acts as an input to the MMR, QCNN, and the QVT algorithms. Our main goal is to extract the best features of the models to make a final model that increases the diversity for different kinds of tumors, since the MMR's principle is based on the analysis of shape and structure, and on the other hand, the QNN and QBTs are based on classification to recognize patterns in more complex data.
Skull stripping is a vital pre-processing step when analyzing brain MRIs for any purpose, including for tumor segmentation. So in MRIs, the skull and intracranial tissues, which aren't part of the brain, have the same bright intensity as the brain and its tissues. So it makes it harder to segment the brain if you don't first isolate it from the skull. Also, parts that do not belong to the brain add a lot of unnecessary noise and detail. So to skull strip, the image was first binarized with adaptive Gaussian thresholding. Then two images were created to overlay on each other to determine the final edges for contouring. One mask was created through erosion, so taking away at the edges and removing noise, and the other through dilation, preserving details and general shape. Contouring is plotting the outlines of segmented images. We overlaid the two masks by weighing them differently with a 70 and 30 split, and it created an image with a good middle ground, enough edges and gaps, thus maintaining the original picture's general contour with a good level of detail and not too much noise. Then the contours of the weighted image were blurred and combined. When the median blurred contour images were set, we identified the wanted contour by calculating the contour areas and choosing the largest one, which would be the brain contour. The contour was used to make the final mask and was applied to the original MRI image, producing the final brain stripped image ready for further preprocessing. Improvements on the skull stripping were implemented to help it run on MRIs of different orientations, lower contrast and quality, and tumors on the edge of the brain. These tumors are of much greater intensity than the brain, so when they're on the edge, the skull stripping originally segmented them out too. As shown in these pictures right here, the skull and intracranial tissues, or the border surrounding the brain, is removed in all four of the results on the bottom. Removing the border helps with the tumor detection, as in most cases, the tumor has the same color gradient as the border, which makes the MMR and QNN algorithm more efficient in processing and spotting the tumor. We are aware of all the complexities associated with brain MRI scans, and we're also working on further developing this pre-processing step. The next step in our algorithm is the mathematical morphological reconstruction algorithm. We have gotten results for the MMR with an accuracy rate of 92% through repetitive ratio testing. MMR enhances and extracts image structures by iteratively combining dilation, erosion, and watershed thresholding operations through its iterative process. Dilation enlarges regions in an image, whereas erosion erodes the boundaries of regions in an image. The algorithm removes noise and artifacts by creating the marker image, which represents key points in a mask image. Based on the ROI and morphology operations for noise removal. Finally, watershed thresholding and segmentation is applied. Based on the concept of segmenting the ROI through a watershed line. Finally, process and results. The creation of marker and mask images guides the four-step reconstruction process. After fine-tuning noise removal with a peak signal-to-noise rate of 39.8, subsequent morphology operations are deployed. The integration of watershed thresholding and segmentation yields a 92% accuracy rate in early stage tumor detection. These are some of the detection results from the MMR algorithm. The tumor is circled here in red, and the size and location are visually accurate. To find the accuracy rate, the ratio of the values taken from 0 and 1 equals true yes tumor, and 0 equaling false yes tumor. So we plan to add this algorithm to our model because of the tumor diversity in our dataset. As MMR is a less strong algorithm that doesn't go through expensive computation compared to our deep learning algorithms, we will use these scans with more vibrant tumor gray ranges for this model.
So the next part is the QNN algorithm, and at our stage in this research, we're currently finishing the process of training and soon we'll be testing this algorithm. So to get an understanding of the QNN, we'll first discuss the classical convolutional neural network, or the CNN. So we'll be applying an architecture of the classical CNN in a quantum environment. The CNN is a deep learning algorithm that feeds training data and detects patterns to process and classify our dataset. A classical CNN will consist of an input, some hidden layers, as you can see here, and an output layer. So for example, here there's a few hidden layers, but you can customize these towards your goal. And in this image, you can see that there's different kind of layers with an input as the cat and the output as classifying whether there is a cat or a dog. You can also see that the layers take different perceptrons. So these circles are perceptrons which take an input and give an output, and the algorithm's learning process essentially adjusts these weights to detect patterns. And if you take a closer look, this specific image has two hidden layers, so two layers with perceptrons. But in the previous image, there were more layers. As I said, you can customize it to your liking, and that's what we did for our algorithm. So our QCNN will use classical data with quantum computing, meaning our quantum convolutional neural network will use a combination of quantum computing concepts with the CNN that we just discussed. So like the classical CNN, we'll have convolutional and pooling layers, but we'll use qubits and quantum circuits instead of pixels in the image. So as you can see in this image, we plan to apply alternating convolutional and pooling layers on our quantum circuit. And the secondary purpose of our research is also to test if the QCNN has a better accuracy or speed than the classical CNN. So the QCNN algorithm will contain three main parts: the encoding, processing, and then the measuring of the data. So first, the encoding consists of the classical input data being converted to a quantum circuit through angle encoding, which just puts the qubits into superposition for parallel computation and applying rotation so that the data can be further processed. So next is the processing step. So like the CNN's hidden convolutional and pooling layers, this applies, it's very similar, but it applies gates and entanglements with specific parameters. The convolutional layer would involve the same feature extractions that a classical CNN has, and then the pooling layer would reduce the dimensionality of the quantum circuit while keeping the same data. So to do this, we'll encode the information from one qubit to the other qubit, and then once we do that, we can disregard the original qubit for the rest of the process. So for example, as you can see in this image here, the data from Q0 is encoded into Q2, and then the data from Q1 is encoded into Q3. And then towards the end, we can disregard both Q0 and Q1, and we're left with just Q2 and Q3, since all the data is stored in those two qubits. And this pooling would just reduce the number of qubits in our circuit by half, in this case from 4 qubits to 2 qubits, and we would continue to repeat this until we end up with 1 qubit, serving its purpose of a smaller representation of the data without removing important information. And then lastly, we would measure the output of a single qubit through a softmax layer, which forms a probability of a specific state, which would then be translated into what that means classically, so depending on whether or not there is a brain tumor.
So the last algorithm of our model is the quantum vision transformers. Right now, we're working on training and testing. So before we delve into the quantum aspect of this algorithm, let's first analyze the classical vision transformers. Vision transformers are an image processing neural network-based architecture that has recently grown quickly by combining powerful techniques such as self-attention, allowing each element to make relationships with other elements to the comparison of significant elements for spatial information. The purpose of the QVT in our model is to overcome the challenges of the QCNN, such as capturing global relationships, increased scalability, positional embedding, and being restricted by a certain number of layers. We can go through the VT's process by referring to this layout. Firstly, vectors are extracted from the image, then split into 16 by 16 patches. These patches get embedded through linear projection. The purpose of these patch embeddings is for parallel computation and for retaining positional data, which will then be processed by an encoder. The vision transformer encoder contains two main layers, the multi-self-attention layer and the MLP head. Before and after these layers are normalization and feed-forward layers, respectively. The purposes of these multiple layers is to make residual connections, prevent overfitting or generalizing new data, and data loss. Before analyzing the multi-self-attention layer, we need to know what self-attention clearly is. Self-attention is a mechanism in neural networks that allows the model to weigh all the aspects of the input, not just in localized regions like in the CNN. This means that the VT will compare every pixel with respect to every other pixel in the input. The importance of these inputs are then stated in attention scores. Delving deeper, this concept is represented in the encoder through the query, key, and value parameters that calculates attention scores, respectively compute, compare, and represent each element in the sequence. The model splits the parameters n ways and passes them into the attention head. Through the linear layer, these parameters contribute to the score by calculations with the information of positional embeddings found earlier in the linear projection layer. At the end of the cycle, information from the query, key, and value parameters go into a single matrix for an attention score. After going through the normalization and feed-forward layers, the data then gets processed into the multi-layer perceptron head, or MLP head, to introduce nonlinearity to refine relationships within the data. The attention score goes through a series of linear transformations and nonlinear activation through a ReLU layer. The transformed attention score goes to the masked self-attention layer, or the decoder, where the model is trained to differ from the masked patches versus the normal to prevent overfitting. The transformed attention score goes to the classification head to get the result if a tumor is present in the MRI scan or not. So now that we understand the vision transformer's classical aspect, let's delve into what quantum could possibly contribute to the VT. For the QVT, we're adapting a shallow quantum circuit, which is a quantum circuit with minimal depth, which could be used in the multi-self-attention and MLP head layers in the encoder. Specifically, we'll be focusing on data loading methods, such as inputting a quantum feature map and compound matrices, and both of which provide the advantage of runtime and the number of model parameters. And aside from data loading, we're adapting quantum orthogonal layers, which replaces the MLP layer for quantum applied nonlinearity. In the first image, the patches are loaded into the qubit states, and then they're processed to the quantum orthogonal layer W, and additional variations of the data, like its inverse, are then applied to prevent overfitting. And this layer creates a probability state, which helps preserve the circuit data through more matrix computation.
And finally, in terms of future work, our next steps include completing the training and testing for the QCNN and the QVT algorithms to compare these results to the CNN algorithm and the MMR algorithm to see if there's any noticeable advantage in terms of speed or accuracy. And then finally, future possibilities to expand this project further down the line would be to classify types of brain tumors rather than simply detecting them, which could affect the action speed for doctors to initiate effective instruction at an early stage. And lastly, we would like to thank our advisor, Dr. McMahon, who has guided us throughout this research. And we would also like to thank ASDRP for giving us the opportunity to conduct this amazing research. And next, we want to thank Adelina, who was a researcher from another group last year who introduced us to quantum computing, that aspect of our research, which was really helpful for us. And then finally, of course, the rest of the researchers on our team who have contributed throughout. Here are our sources. Thank you. Does anyone have questions?